Hi guys, welcome to the revision lessons on Unit 4 Biology. In this lesson, we're going to be covering mutations and chromosome abnormalities, and this is under Area of Study 1, Outcome 1, How are species related. So specifically, we are going to be talking about the qualitative treatment of the cause of changing allele frequencies in population, including gene pools and types of mutations such as point mutations, frameshift mutations, and block mutations, and how these form new alleles, as well as chromosome abnormalities like aneuploidy and polyploidy. So by the end of the lesson, you should be matching your knowledge against this dot point to ensure that you've understood the particular content for the relevant part of the study design. So in a population, we have alleles, and alleles are known as variant forms of a gene. As you know, the frequency of alleles can change over time, and this is through a number of mechanisms, and we're going to be looking at these two today: mutations and chromosome abnormalities. These will be covered in the following lesson. So the types of mutations that we have, there are point mutations. That's a nucleotide substitution. So for example, you have a 3 base pair. One of those bases will be altered, and we have now a CCT. A frameshift mutation, we have deletion or insertion of a nucleotide into the sequence. That'll change the reading frame or the way that the protein or the amino acid is transcribed and translated. We have block mutations. That's typically in chromosomes, where large segments of a gene or large segments of a particular part of the DNA are changed. So it's a block of nucleotides. We also have aneuploidy and polyploidy. That talks about changes in chromosome number. The whole chromosomes are changed. So point mutations, specifically, there are three subsets. We've got silent, we've got missense, and nonsense. You need to know these. So a silent mutation is a change of a nucleotide base, and we have no resultant change in the amino acid. So here is our mRNA that's been obviously transcribed from our DNA. If we have a silent mutation, you can see that one of the base pairs over here is changed. It's also shown in red, and there's no difference in the amino acid that's produced. So that's a silent mutation. No change to the amino acid. A missense mutation is where we got our nucleotide sequence here, and we've changed one of the amino acids. That will change, so we've changed one of the nucleotides, and that will result in a change in the amino acid. From there, we also have a nonsense mutation. So nonsense mutation is a changing of a nucleotide base, which results in the production or premature stop codon. So over here, we have a change in the amino acid from the A to the U, and that's a nonsense mutation, meaning we'll have a stop codon produced, meaning the protein will no longer be translated. They're point mutations, changes in one nucleotide, which will affect the protein in 3 different ways. So here's a summary of just the point mutations shown over here. Frameshift mutations. So this is the insertion or deletion of a nucleotide, and that alters the reading frame of the protein, as I already mentioned. So here's our original sequence. You can see here we've changed, or we've removed, in this case, we've inserted 2 different nucleotides, and they will result in the production of another amino acid, therefore changing the reading frame. We no longer produce alanine as the second amino acid, we produce glutamine. And now leucine is the third amino acid, and arginine is originally the second. You can also obviously have deletions as well. So mutations in chromosomes are known as block mutations. So there are many different types. We've got deletions, where you'll delete parts of the chromosome. You have duplications, where obviously the opposite, you duplicate parts of the chromosome. Inversions, where you flip around or swap. So you can see here the banding pattern from the bottom is now the banding pattern at the top. We have insertions, where parts of the chromosome are inserted from each other. So this chromosome here, chromosome 20, is being inserted into chromosome 4, and we have chromosome 20 there. We also have translocations. So translocations is where one part of a chromosome, they swap with each other, and now we have a combinant chromosome. So we have chromosome 4 and chromosome 20 pairing together in this instance. So now chromosome abnormalities and how they change allele frequencies. Aneuploidy is the first one of those. That's an addition or a deletion of an entire chromosome, and it's altering the diploid number of that organism. So for humans, we have a diploid number, 2n equals 46, which is 23 pairs of chromosomes. You can see over here, this is a normal human karyotype in A, so normal human karyotype here, and this one here is a human karyotype which shows only a single sex chromosome. So we don't have a Y, we're missing the Y, we have the X chromosome. And there was not another X, so it's not a female, it's not a male. It'll be a female, but they're obviously missing a chromosome. Polyploidy, that's when an organism has multiple sets of chromosomes. So they could be triploid, or they could have 4 times the amount of chromosomes. So you can see every chromosome here, there's 3 sets of each of those chromosomes. That's known as polyploidy. So this one is triploidy, is a specific example. This is the summary of the ideas. So we change allele frequency in populations through mutations. So if we change point mutations, we have point mutations, we have frameshift mutations or block mutations. From there, that will alter the allele frequency in the population, and that may result in evolution. Key terms, so this is what you should know by the end: alleles, mutations, and the different types of mutations, as well as aneuploidy and polyploidy. This is what you should be able to do at the end of this lesson. So now you should be able to draw and describe the different types of mutations in an organism and how it affects amino acid chain specifically, and draw and recognize differences in chromosome number, such as aneuploidy and polyploidy. Thanks for listening.